Okay, so we're going to be talking about microscopy today. First thing is our cool science thing of the week. In this case, it is imaging with subcellular dynamics in multicellular organisms. Now, what this actually means, because it's a very long title, uh, it's combining two different types of microscopy to visualize cell types really, really rapidly. So normally when we look at a microscope, we're always looking at like one little slice. We're not looking at a giant overall picture. And because a lot of things in nature are three-dimensional, that limits how well we can focus on things. And we can take multiple slices. The problem with that is it's very slow, it's time consuming, it doesn't give great resolution. And then another part of it is we can't really see through these things very easily. So even if we are taking slices, it's really slow. So we can't see things that are like living, that are like moving around. We usually have to kill them or hold them in place, which is not great, especially if we're trying to study living things. So these researchers, they're at the Howard Hughes Medical Institute, came up with a new type of microscopy combining lattice light microscopy and adaptive optics. And this allows you to visualize organisms real time. So if you see, you can look at the blood vessel here that's imaged. It's actually really, really cool. It's 3D, but it's done in real time. And that's what makes it really, really amazing, because a lot of this would normally have to be taken over several minutes and things might change. This is done in real time and it creates a full 3D image rather than just kind of like a faux 3D image, which we're usually used to. And the reason why it's so cool is because you can see it can visualize multiple different colors at the same time, so different fluorescences. So here we can see a zebrafish eye, we can see tight junctions, as well as I believe those are stained for the rods on the membranes, which is actually really, really neat. This little purple picture here, that's a blood vessel, and the green stuff is a tumor actually infiltrating out of the blood vessel. So that's actually really cool. And the multicolored image here is a neural crest from a zebrafish, which is actually very, very neat to see and not something that we can typically see in real time. We can actually visualize it on here. And so you can see it's actually a cool little instrument. This is a very first run type of thing. And you can see it's basically kind of held on with zip ties and pegboard. It's not super fancy in terms of setup. It's not a pre-made device. So it's actually kind of hobbled together by scientists. So that's actually really cool about it. Now, what are some of the limitations of this? Well, even though it gives us really, really great images, one of the major limitations is it gives us huge amounts of data. So like a minute of recording is like 0.62 terabytes of raw data, which gets doubled. So there's even more data because then they have to run it through the imaging process, the adaptive optics, which take up even more power. And you have to do it with processing to make the 3D video. And that can take several days, several hours for a short one, several days for very long or complicated ones. And they actually have their own like little rendering farm to be able to do this. So it's very time consuming. You also have to use organisms that you can see through. So zebrafish embryos are what they use a lot because we know a lot about them, but we also can see through them, which makes them convenient. So there are some limitations to it. It's not a perfect technique. But you can see some of the images are really, really stunning. In the video, you can see a white blood cell moving through the ear. Now, we've never seen that before because we can't image that live. But we can image it live here because we can see through the layers of this organism into the ear and watch it move around. We can track its centroid volume. In one of the images here, you can actually see cell reproduction going on in real time, stained. It's actually very, very good. And the images are so high quality. And of course, you can turn it and continue the movie whatever you want, because it's a three-dimensional image. It's not just a static image. You can look at it from every conceivable angle. You can push through things. You can look through different layers. It's great. It's very, very neat. So I highly recommend looking through the video yourself. So whenever we talk about microscopy, you want to look through the objectives to kind of figure out what we're doing. They're also going to be part of the basis for your questions. They will be involved with the next quiz coming up. For the microscopy lab, there are some safety things, primarily making sure you're holding the microscope properly. Make sure you grab the microscope on the arm and put your other hand on the base of the microscope. So you're always carrying it with two hands because they're very heavy and it's easy to bump them or drop them, and we don't want to do that. Microscopes are assigned by your lab seat number. So if you open up your center drawer, there's a little number in there and you grab that same microscope. That way we know who is working with which microscope so we don't take somebody else's microscope. Also with the slides, be very careful with them. Some of them are finicky and old. They're also all made of glass, so if you break them, they can cut you. So be very careful. Make sure you guys also place them back in the same box that they belong in. Don't just take random slides. Make sure you guys put them back in the same exact spot and nice and evenly so that we can keep everything nice and tidy. Also, make sure you guys clean the microscope lenses each time between slides. So whenever you put oil on or just use it in general, sometimes it gets the lenses dirty. What you want to use is a little tiny piece of optical lens white paper that can be found in your center drawer. It is different than bibulous paper, so don't confuse them. Make sure you understand the difference. So optical lens white paper is only for optical lenses. And you just tear off a little tiny piece and you can clean the lens. in between each one, and make sure you do it at the end of each session as well. Also, a quick reminder, make sure you guys only use the fine adjustment knob on 40 and 100x lenses, because coarse adjustment knob will move the stage up rapidly and it can accidentally shatter the lens. We'll talk more about that in a second, but make sure you guys only use one. All right, parts of the microscope. So you guys should know the parts of the microscope. You should also know who came up with the first modern type of microscope, which is a compound microscope. That's Zachariah Hansen, 1600s. So it's been around for a while, but our modern microscopes are significantly better. But they still follow kind of the same principal ideas. And the first part is that we have an optical lens, an ocular lens. So that's where we look through with our eyeballs. It also has its own magnification power. So these lenses actually have a 10x magnification to them. We also have the body tube here where the light is pushed up through the ocular lenses. The arm is the mounting part for the body. That's the part that you hold on to when you carry it. At the bottom, we of course have the base and the voltage control. This is also sometimes called the rheostat. And that controls how much electricity is going to the light bulb to increase the light or decrease it. And of course, over here we have the coarse adjustment and fine adjustment knob. Coarse adjustment moves the stage, which is this guy right here, up and down a significant amount. And then the fine adjustment is dialing that in. It moves it in very, very tiny amounts up and down. Also over here we have the objective lenses. You can see they're attached to this rotating nose piece, so they go around kind of in a circle, so you can look at each one individually. Below the mechanical stage, we have this thing called the condenser. It does exactly what its name implies. It condenses light through your sample, so you see more light coming through. And of course, we have the light source at the bottom, which is a light bulb. Underneath the mechanical stage and right underneath the condenser, you can see this other dial. That is the iris annulus diaphragm. It is exactly what it sounds like. It is an iris. It opens and shuts. It helps sometimes visualize bacteria where you have a hard time kind of distinguishing the edges or getting a really sharp image. If you play around with that a little bit, it works out great. The other settings for the condenser we're not going to touch, so make sure you guys leave those alone. On the other side, you can see these two little knobs right here. This moves the mechanical stage forwards, backwards, left, and right, and so it lets you move your sample around on the stage so you can visualize it different areas. Ocular adjustments are really, really important. So because we have two ocular lenses, we can see two distinct images, and if you're not having them set properly, it'll just come up as two little circles when you look in. However, what we want to aim for is one large, even circle. So we have to pull it apart or push them together, depending on how far your individual eyes are set apart. And of course, these are shared, so it's going to be disrupted every single time you use these the next day. But you also need to keep your eyes about a certain distance away from the ocular. So obviously don't have your eyeballs touching the lenses, but maybe like a couple centimeters away, and you just kind of get used to holding your head at that height. Eventually, you just get used to playing around with everything to make it one large image, but that's the way you want to see it. So if you're not seeing this one large circle when you look through the microscope, you need to do some adjustments to make it work properly. Now, focusing microscope, very tricky for the first time, but you guys can get it. It is an important skill to have, so make sure you guys work hard on that. The way we focus, so whenever we're focusing, we're looking at kind of a plane or a slice of the thing that we are looking at. And because even bacteria are three-dimensional, we can put parts of them out of focus and other parts in focus. This is a cyanobacterium, so it's very, very large. Right now, we are focused above this bacterium, so you can see that we're really far above the height. And as we move down, we can see that a good chunk of the bacteria is still in focus, but some parts are still a little hazy. And it's because we're moving into the plane of focus that optimally gives us the best picture. And as we continue to focus through it, some other parts now become even more fuzzy, and we're looking at the bottom plane of this. So there's kind of a nice Goldilocks range you want to aim for when you're looking through a microscope and you're trying to focus. We're always going to start out with a 10x objective on the stage, so that's the one that we want to use the most. Generally, we want to put the stage as far down as we can go. We're also always going to skip the 20x. That's worthless for us, so don't use that. We put the slide on the stage, make sure it's right side up. That is the side with the cover slip or where the bacteria are. You're going to then use the coarse adjustment knob while you're looking through the oculars, and you're going to wait until you see something kind of flash into focus that you can see that looks like bacteria. Once you see something kind of flash into focus, you want to dial it in with the coarse adjustment knob as best you can, but then switch over to the fine adjustment to bring it really, really sharp focus. Now, if you notice, when we get into focus, there's a whole range of area that's actually really, really good for us to see. We might say this is good enough, but when we go up to the next power, in this case 40x, what you'll see is that there is a small area that we can be in focus and it'll carry over to perfectly in focus on the 40x. But if we're just a little out of focus on the 100x lens, on the previous lens, then when we shift over, all of a sudden everything's out of focus. Now, if you notice, I've been talking about 100x and 400x, but the lenses themselves are only called 10x and 40x. Well, remember, this is the total magnification. So our oculars also have a power magnification, which is 10x. So we just add a zero onto the end of everything that we're talking about. So if we're talking about a 10x lens, it's a 100x magnification. 40x lens, it's a 400x magnification. And lastly is the 100x lens, which is a 1000x 10x magnification. And the same thing is true here. So if you go up to the next power, you are even more likely to be slightly out of focus, so you'll have to do some adjustments there. The way you adjust it, you click the 40x nose piece into place. You're going to use the fine adjustment knob only. Do a quarter turn forward, quarter turn backwards. Whichever way is better, keep going in that direction. If neither way is better, you can go up to a full half turn or up to a full turn. But once you get to that full turn, you maybe want to start back on the 10x to get it into sharp focus. Then after you do the 40x, you're going to go to the 100x lens. And this is a tricky part because 100x lens needs oil to work properly. So what you'll do is you'll click it between the 40x and the 100x. We'll have little glass containers with a glass dewar rod that has immersion oil. We will take a drop of immersion oil, we'll put it right in the center of the slide where the light's coming through. Then we'll click over the 100x lens, which is 1000 magnification, and let it sit there for a second. Once it comes in direct contact, so any kind of bubbles and stuff work themselves out, we will then be able to focus on a object. Now, when we do add in oil basically helps lower the light diffraction index. So once we basically get a really, really good amount of light going straight up in through the microscope, then we can see it. So if we didn't use the oil, we wouldn't see anything. It would just be very hazy. We add in the oil, it becomes much sharper. You'll again have to use the fine adjustment. And a lot of times when you go to the 100x lens, you're going to get kind of really out of whack because of the way the oil works. So you're going to have to do some definite adjustments going from 40x to 100x. Another thing that's also really important is making sure you don't go back to the 40x lens, because if you go back to the 40x lens, you'll drag oil onto the 40x lens, and that makes it very difficult to use, especially if you didn't clean it properly. So once we've got it on the maximum magnification, and we always want to do that because why not use the most powerful thing that we have, we can look at the bacteria. Now, if you notice here, there's a difference between what is good to look at and what is bad to look at. If there's a bunch of bacteria in a very dense area that's really difficult to see, we can't tell things about shape or arrangement very easily. It's very, very difficult to analyze it. It doesn't work out very well. Instead, if you look on the good side, we see more white space than bacteria. This gives us opportunity to see the arrangement, the shape, and we can see these consistent patterns because they're more spread out. They're in their natural arranged state rather than a giant clump. So always make sure you guys look for more white space than bacteria. So if we're talking about blood smears, which are actually really easy to see because human blood cells are much, much bigger than bacteria, so they're easy to focus on. It was first created in the 1900s by James Wright using a bunch of different types of dyes. You guys don't really have to know those, but you do have to know that we've had this technology around for about 100 years. The way you do it is you put a drop of blood on a slide, you drag another slide across, and it creates this line, and then you push it across and basically smear the blood across the slide. There is a trick to looking at it here. You always want to make sure to look at an area that's not at the very end of the slide, but kind of halfway towards the end of the streak. So in this little tiny area right here, if you look too far to the left, you'll see shredded blood cells and it won't really make a lot of sense. It won't look very good. If you look farther to the right, you'll see big globs of things that don't really look like blood cells at all. So if you're not seeing things that look like this image, then you should try to hunt around elsewhere to try to find a better image because this is what it should look like. And you can see these cells are pretty neat. They are erythrocytes are mostly what you can see. Those are the little donut hole things. They are red blood cells. They are one of the only cell types that do not have any DNA in them, so that's why you can see through them. And they don't have any of the other stain, which is the purple stain there, that stains nucleuses. And these other cells that have the purple stain, these are white blood cells. And we have a bunch of different types here in this image, and we can go over each one. So you should be able to identify these. The first one is a neutrophil. It is a multilobate white blood cell. You can see it's got these little connected parts that kind of look like dumbbells strung together in a line. And then it has a good amount of cytoplasm in the middle. That's just a cell group that's inside of a cell. The next one is monocyte. They tend to be bean-shaped, large nucleus, and large amount of cytoplasm cells. You can see that they are very, very large nucleus and lots of cytoplasm in the center, but they often get confused with lymphocytes. Lymphocytes also have a very large nucleus, but it's very, very round. They tend to have a tiny little crescent of cytoplasm, as you can see here in this image. They don't have that much cytoplasm, so that's how you differentiate them. Lymphocytes are very round, very little cytoplasm in a crescent shape. Monocytes, large, bean-shaped nucleus, lots of cytoplasm. Now the other two types of cell types you see here are basophils and eosinophils. Basophils are base-loving dyes. They like to take up basic dyes, so that's the name, basophil, base-loving. So they pick up purple dye here, in this case, and it attaches to their granules. So they get these nice gritty purple coloration to them, and that helps distinguish them from their companion cell type, which is eosinophils, who happen to like eosin dye. So they also have bilobate nucleuses, just like basophils. Basophils are also bilobate. Eosinophils also have gritty, granular bits to their cytoplasm, but in this case, they're pink. Now, these are harder to find, basophils and eosinophils. They represent a very small amount of blood because usually they're located in specific tissues, mostly the cells that we think about that are related to allergies or more historically to parasites and things like that. So they're usually tissue specific, but they can be found around in blood. There's something like 0.4% or 0.1% of blood, depending on which one you're talking about. So it's very low. One of the other things that you can see in blood smears is platelets. Platelets are the little flecks that you see all around next to the red blood cells. They're pretty easy to identify as well. Now, if we're looking at bacteria, you should know about shape and arrangement. Shape's actually really easy. There's only 3 major shapes that we are concerned about, which is cocci, which is perfectly round bacteria, bacilli, which are rod-shaped, so they're a little squished in the center, and spirali, which are kind of squiggly shaped ones. They've got an S curve to them or a little spiral curve to them. And those are the 3 major types of shapes. Now, arrangement is a little bit trickier. So the first arrangement we see here, which is stolen directly from your book, is strepto, which is long chains. This can be either cocci or bacilli, it doesn't matter, but they're just end-to-end in long chains. You can see these nice long chains here. You can also have diplo or pairs, so you can see multiple repeating patterns of pairs throughout this image here. A lot of bacilli will do this, but sometimes cocci will do this as well. Usually that's a sign, if you find cocci pairs, that it might be a pathogen. The last one here is staphylo on the top, and staphylo is grape-like clusters, so they're little packets of them. This is typically only going to be cocci. You're not going to find this in bacilli. So staphylo tends to be cocci, and you can see it's this nice little grape-like clusters all clumped together. Now, if you look over here on the bottom, we can see tetrads, which is also only cocci, but you can see it's got a repeating pattern of 4. 4, 4, 4, 4, 4, 4. And if you were looking in an area where it wasn't more white space in bacteria, this is a great example of where arrangement becomes very difficult to tell. You might look down here and you might go, oh, this looks like staphylo because they're all clumped together. But if you spread out to a more white area only spot with fewer bacteria, you can see this repeating pattern of 4, which is not natural. This is something that is happening not by random chance, but by the actual process of the bacteria to create these tetrads. So make sure you guys keep an eye out for things like that. Lastly is palisade. Palisade is an uncommon arrangement that we won't see very often in lab, but we'll see a couple times. If you guys think of like a log fort wall, where they have those logs placed in the ground with little tips on them and they're lined up like that, that's a palisade wall. These are palisade bacteria. They line up long ways along here. You can see the example here and here. But they also like to stack on top of each other like you just dumped a big pile of logs on top of each other. So they tend to have a lot of 3-dimensionality as well. So you kind of have to keep an eye out for that one because it gets a little confusing, and you can see them stacked kind of all on top of each other. Bacillus subtilis is one of the bacteria that we're going to be looking at. It's gram-positive. It's also got this long rod shape. It tends to also form in this strepto arrangement. See these long, nice chains here. It's considered a BSL-1, so we generally think of it as the term is generally recognized as safe. We say it doesn't really pose a threat to humans. However, it is actually interesting because it does pose a threat. So if we think about endospores, spore formation is actually very good at triggering your immune system, and it becomes an issue when we're talking about implantable devices into humans because spores are so proliferous in the outside world, they can actually get implanted with the device and cause your immune system to react against implantable devices, which is why we need things like gamma radiation to help wipe them out. Otherwise, your immune system is going to start attacking them. So even though they're generally safe, they do have potential to cause disease in humans under very specific conditions. And that's not really disease so much, but as a reaction in humans. Staph aureus is another one that we're looking at. We consider this BSL-2. It's found in the skin of mammals and also in their nasal cavities. It can be considered normal flora to an extent. Some people just have it on them normally. We can also think of it related to certain diseases. Now, one you might be thinking of is MRSA. It's not actually a disease. It stands for methicillin-resistant Staph aureus. So it's just a type of Staph aureus, but it's commonly associated with different types of infections. It can be like wound infections or systemic infections, things like that. You can also get different types of diseases from them. A common one is scalded skin syndrome, which typically happens to children or neonatal babies because their immune systems aren't really set up to handle it yet. And it'll pick up a toxin that makes their skin exfoliate and look kind of like it's been scalded in hot water. You can also get things like toxic shock syndrome and lots of other diseases associated with Staphylococcus aureus. E. coli is another organism we're going to look at. It is a small gram-negative rod-shaped one, so bacillus. Now, if we're looking at this slide, you might say, okay, but this slide is actually a bad slide, and it is because there's more bacteria than white space. But from this slide, we can actually see some of the arrangement here, so we'll keep looking at this one. You can see little packets of pairs running around here. And often when we think about pairs, they also tend to be things we would call like diplo-random because A lot of times it might be diplo somewhat, and then also a little random, so we'll call them diplo-random arrangements. That's fine. We generally consider them BSL1. They're actually part of the normal flora of all mammals, not just humans, but all mammals. But certain strains can cause serious diseases. The ones you guys might know about the most are O157H7, and that's its antigen determinant on the outside. So that's what that name means, and it can cause a serious problem. So we sometimes just consider it BSL2, even though it's mostly BSL1. Lastly is Rhodospirillum rubrum. You guys can see it's got the nice little squiggle shape. So this is a spirili example. And it's got little flagella on there on the end, so it'll be actually very modal. It'll move around. It won't stay in one place. So it's important to keep it. Remember that that's one of the contributing factors to why some things are random and other things only have one certain type of arrangement. This one's random because it's always moving around, and it's that spirali squiggle shape.